Fusion Mobile, quality e-learning experience on the go.
Okay, you're welcome to yet another series on biology. I am Olajide, and this program is power packed by Fusion Mobile e-learning clinic. Today we're going to be looking at the development of new organisms, how new organisms tend to develop from the baby stage down to the adult stage. We're going to be looking at the various structures involved, the various chemicals secreted, and we're going to be looking at everything extensively. Stages in development of toads. Stages in the development of toads. Before now, I would need you to understand that the male organism produces sperm, the female organism produces egg or ova. These two will fuse together and they will form what we call the zygote. The zygote will undergo cell division. This is mating, fertilization. They will form zygote. The zygote will undergo cell division and it will form what we call the embryo. As you need to remember this stage, the sperm comes from the male, the egg comes from the female, both of them mate and fuse together, they form the zygote. The sperm is N haploid, the egg is N haploid, the zygote is 2N, N plus N, 2N. The zygote undergoes cell division and it forms the embryo. So now we're going to be looking at the stages in the development of toad. Toad is a representative family of the amphibians. The stages of development actually start from courtship. Don't forget, fertilization in toad is external. Fertilization in toad is external. Toads in amphibians and fishes. Amphibians and fishes undergo external fertilization. Reptiles, birds, and mammals undergo internal fertilization. So please don't forget this fact. Reptiles, birds, and mammals undergo internal fertilization. Amphibians and fishes undergo external fertilization. So their stages of development start from courtship. After mating is done, egg stage, the egg is fertilized, the egg stage. From the egg stage, they get to the young tadpole. From the young tadpole, they get to the external give stage, down to the internal give stage, down to the limb stage where they develop limb, and then the young toad. The young toad is ready, and that's the young toad stage. Now the hormones that are responsible for this development is called thyroxine. Thyroxine is secreted by the thyroid gland. That's the hormone responsible for all this development aforementioned. Metamorphosis is the next on the list. What is metamorphosis? First, you need to understand that metamorphosis is seen in insect. That phenomenon is seen in insect. It talks about the gradual changes that are seen, are you with me, from the baby stage of the insect down to the adult stage. The gradual changes that are seen from the baby stage down to the adult stage. There are basically two types of metamorphosis. We have the complete metamorphosis and we have the incomplete metamorphosis. We have the complete metamorphosis and we have the incomplete metamorphosis. The complete metamorphosis has got 4 stages. The incomplete metamorphosis has got 3 stages. We are going to be looking at the tabular differences between complete metamorphosis and incomplete metamorphosis. So we're going to be looking at the tabular differences between complete metamorphosis and incomplete metamorphosis. As I've said earlier, complete metamorphosis has got 4 stages. What are these stages? They are the egg down to the larva, down to the pupa, and down to the imago. Imago is also called adult. Now for the incomplete, they have just 3 stages. 3 stages. One particular stage is missing. Now you can get the answer from the second differences that has been presented in this table. And what is it? Pupa. It lacks pupa. So for this one, we're going to be having the egg, the larva. No. Instead of us having the larva and the pupa, we're going to be having, because the pupa is absent, the larva is not going to assume this name. It will assume a name called nymph. The nymph and the imago. Now here we have the egg, the larva, the pupa, and the imago. For that one, the pupa is absent, and so the larva stage is for the nymph here. Are you with me? So complete metamorphosis has got 4 stages. Incomplete metamorphosis has got 3 stages. There is the presence of pupa for the complete metamorphosis. incomplete, lacks the presence of pupa. No resemblance between pupa and adult. The pupa and the adult has got no resemblance. However, the nymph and the imago, which is the adult, has got resemblance. Are you with me? Examples are housefly, butterfly, mosquitoes. Examples here are termite, cockroach, and grasshopper. So quickly we'll be traveling into the world of plants. I will be looking at their reproduction. So now we're going to be looking at the development of new organisms as regards to plants, their reproductive structure, and how new organisms tend to appear or tends to be. So the first stage is pollination. In our previous classes, we've talked about pollination extensively. We said it's the transfer of pollen grains from the anther to the stigma. We look at the cross-pollination and the self-pollination. I would actually advise you to go back and rewatch that video so you have a comprehensive knowledge before you actually get to this place. Are you with me? So after pollination is done, the pollen grain is now transferred from where it was originally to the stigma, and where fertilization will take place. The structure of the pollen grain, we also looked at that. We said the pollen grain has got two coats called the exine and the intine. It has tube nucleus and it has the relative nucleus. So now we're going to be looking at the structure of a mature ovule. It has the ileum. What is the ileum? The ileum is the point of attachment of the ovule to the placenta. The ovule is attached to the placenta via the ileum. The ovule is attached to the placenta via the ileum. The integument are the two coverings. The two coverings of the ovules. The two coverings of the ovules are the integument. The two coverings of the ovules. The two covering of the ovules are the integument. The embryo sac. The embryo sac is where the female gamete is produced. The nucellus has a nutritive function. It is nutritive in function. It provides the ovules with nutrients. And lastly, we have the micropyle. The micropyle is the opening into the ovules. It was left behind by the integument. And so now we're going to be looking at fertilization in flowering plants. Okay, so we're going to be looking at fertilization in flowering plants. As I said earlier, we've got the pollen grain. The pollen grain has the outer coat called the exine, the inner coat called the intine, which gets its constituent from cellulose. We have the germinative nucleus, which is going to produce two male nuclei, and we have the tube nucleus. So what happens is this: the pollen tube, the pollen grain, and not tube. The pollen grain can get transferred by insects, by wind, and when it gets transferred, it lands on the stigma. The stigma has sugary substances. So what happens is that the pollen grain will absorb the sugary substances and it will germinate. That is stage one. It will germinate. So after it has germinated, the outer coat, remember something like this. The outer coat called the exine will disintegrate. It will break, it will disintegrate. So the outer coat breaks. When it breaks, what happens? The pollen tube will now what? The pollen tube will develop into the style. It will develop and it will pass through the style. When it passes through the style, it will get to a point where it will get very close to the ovule. So at that point, listen up, at that point, the end of the pollen tube will burst. Recall, the outer coat breaks, pollen tube grows into the style. So you get to a point where the end of the pollen tube will burst after growing into the ovules. So at this point, recall it has two male nuclei. One of the nuclei, the two male nuclei will actually be released into the embryo sac. The embryo sac is where the female gamete is. It is where the female gamete is. The embryo sac is where the female gamete is. So one of the nuclei, we have two nuclei. One of the nuclei will fuse with the ovule and it will form the zygote. The zygote will undergo fertilization. It will form the embryo. The other nuclei, there are two. The other nuclei, we'll call it the secondary male nuclei. It will now fuse with the secondary nucleus and then they will form the endosperm. The endosperm is triploid in nature. It is triploid in nature. That is 3N. The major function of the endosperm is to store food. So now we're going to be looking at the embryo, the structure of the embryo. Okay, so now we're going to be looking at fertilization in the embryo. Don't forget, it is the zygote that forms the embryo. The zygote forms the embryo through a cell division called mitosis. It forms the embryo through a cell division called mitosis. The cells rearrange themselves and they form the embryo. The embryo contains of several major parts. One of them is called the plumule. The plumule develops into a future shoot. So the plumule at this stage is called the embryonic shoot. It's called the embryonic shoot. But in the future, it becomes a shoot. The part of the plant that is above the ground is called the shoot. The part of the ground that is not seen under the ground is called the root system. So a plant has two systems, the shoot system and the root system. The part that is above the ground is called the shoot system. The part below the ground is called the root system. So the plumule is called the embryonic shoot. It is what will form the future shoots, the leaves, the stem, the branches. Those are part of the shoot. While the radicle is called the embryonic root. It is what will form the future root, embryonic root. It's called the embryonic root. It is what will form the future root. Now take note, the plumule is ensheathed. The plumule is covered. The plumule is shielded. The plumule is protected by a structure called the coleoptile. It is called the coleoptile, where the radicle is protected by a structure called the coleorizer. So we have the coleorizer and we have the coleoptile. The cotyledon is also present. Cotyledon may be one or two, depending. When it is one, we'll call it monocotyledon. When it is two, we'll call it dicotyledon. All grasses are monocot. All grasses are monocot. Don't forget, maize is also a monocot. Maize is a monocot. Rice, millet, and so on, they are all monocot. Monocot has one seed leaf. Two seed leaf, we call them dicot. A very good example is a mango, orange, etc. Good. Lastly on our list, we have the endosperm. The endosperm is a structure that stores food. The endosperm stores food. It stores food. And most especially grains have got endosperm. When you see your rice, there's a particular structure. If this is the seed, this side, it stores food inside. So we call this place the endosperm. Don't forget, now endosperm. And lastly on our list is the endospermic seed. A seed that stores food. A seed that possesses endosperm storing food is called endospermic seed. Don't forget, you can go through this particular point again to get a real idea of what I'm explaining. So now we're going to be traveling into the world of germination. Okay, so we're going to be looking at germination. Germination are the series of changes that involve the transformation of seed into seedlings. We have seed and we've got seedlings. The transformation or the changes, they are called germination. They are called germination. So let me explain something to you that you need to know. Seedlings is not the end point of the growth of a plant. Seedlings are the part of the plant that you see. I planted a mango seed. When I see the mango growing, we'll call it seedling. The seed is under the ground, underneath the ground. When you see them growing, you call them seedlings. And mostly we call those the plant growing seedlings when we see them having 3 leaves. We call it trifoliate. When they start growing 3 leaves, we say they are what? Seedlings. Take note, these leaves will still grow and become trees, shrubs, or whatever. Are you with me? So the part of the plant that when you see the plant growing and plant has 2 or 3 leaves, we say they are what? Seedlings. But the seed is under the ground. Are you with me? So when seed get to seedling, germination stop, then growth will resume. Germination is also a subset of growth. However, plant has to grow, secondary thickening has to come in and all that. I will still be looking at that. So germination are the series of changes involving the transformation of seed into seedlings. The embryo of seed tends to change to seedlings. I will establish that. Now, most seed undergo dormancy before they grow. At times, we plant our bean seed and under 3-4 days, nothing is coming out. And we're like, this seed is dead. Hey, we have been duped. Stuff like that. So, the period of rest undergone by seed is called dormancy. Dormancy is the period of rest. The seed is still alive, but their physiological activities has been minimized. Are you with me? It has been minimized. So, they are resting. They are trying to gain vigor and strength to continue the growth processes. And what are the types of germination? There are basically 2 types of germination. Basically 2 types of germination. We have the hypogeu. Let me change the marker. We have the hypogeu germination and we have the epigeu germination. Hypogeu germination and we got the epigeu germination. Hypogeu germination and we have the epigeu germination. Hypogeu germination. Let me write it again. Markers somehow. Hypogeu germination. And we have the epigeu germination. What is the meaning of hypogeu germination? What's the meaning of epigeu germination? When the cotyledon or the seed leaf is under the ground, we'll call it hypo means below. Epi means above. Gill here talks about the cotyledon or the seed leaves. When the seed leaves, the cotyledon, when they start growing under the ground, when their growth region is underneath the ground, we call it hypogill germination. But when their growth, the growth of their leaf is seen and is above the ground, we call it epigill germination. Hypogill germination is seen with monocot. Monocot, grasses generally, monocot grasses, maize, rice, millet, all those grains. We call them monocot. But epigill, their seed leaves are above the ground. They grow, they are offshoots. They grow above the ground, above the soil, not below the soil. We call it epigill. And dicot undergo this. Dicotyledonous plant undergo this. Example is our mango, our beans, our cowpea, vigna, guttilata, and every other legumes that you could think of. So those are the types of germination that we got. What are the conditions necessary for germination? Water or moisture must be there. Oxygen or air must be there. Enzymes must be there. Energy in form of food must be there. And lastly, viable seed must be there. A viable seed is a seed that is alive. It's not a seed that has been damaged by insect. It's a seed that is alive. Now, what are the essential factors for the development of organisms generally, whether plant or animal? Food must be there. Oxygen must be there. Water must be there. Warmth must be there. And good health. Good health. Good health. Health is wealth. Health must be maintained and guaranteed. Okay? So now, we're going to be looking at germination, not germination, so we're going to be looking at development in animals. Starting from the womb stage, we're going to be looking at the placenta, we're going to be looking at the uterus, the embryonic membrane, amnion, allantois, everything we're going to be looking at it. Okay, for the purpose of this class, we're going to be looking at two categories of animals. We're going to be looking at the egg-laying animals. We're going to be looking at the egg-laying animals. Oh, let me change this. We're going to be looking at the egg-laying animals, and we're going to be looking at let's just call it the non-egg-laying animals. Okay, the non-egg-laying animals are mammals. They don't lay egg. The egg-laying animals are P, A. The purpose of this class, we're going to be coding it P, A, R, B. So P for Pisces, which are fishes. A for amphibians. R for reptiles, and B for birds. So it's only mammals that do not lay egg. Mammals that don't lay egg. It's just for the purpose of this class, because when you get to like higher level, you get to see that there are different categories of mammals as well. There are mammals that lay egg. There are mammals that put their baby in pouches. And the mammals that will allow the child to be totally developed before they give birth to them. But for the purpose of this class, we have just egg-laying animals and non-egg-laying animals. Are you with me? So the egg-laying animals are Pisces, those are fishes, amphibians, reptiles, and birds. So their eggs, what happened to their eggs? When the egg has been laid, fertilization is done. Take note, for fishes, fertilization is external. For amphibians, fertilization is external. Reptile, birds, and mammal, fertilization is internal. So for their eggs acquire shell. Their shell is made up of calcium. It is a protective shield for the egg. They also acquire what we call membranes. They get what we call membranes, and they acquire egg spaces, and they acquire albumen. The albumen is called the egg white. The egg white, if you take egg, that whitish part is called the albumen. It constitutes 50% of the body, and they have what we call the yolk. The yolk is the inner part, and lastly, what we call the chalaza. Chalaza are the protoplasmic strands that are present therein. So in mammals, mammals have got uterus. The uterus is the womb where the baby is actually implanted, but now we don't call it baby because it has not been given birth to. We call it embryo. So the embryo is implanted in the uterus. Fertilization takes place in the fallopian tube. While the embryo is implanted in the uterus, we call that process implantation. While the placenta connects the embryo to the mother. If the mother, they take oxygen from the mother, pass it down, they take blood, all needed material, take it from the mother and pass it down to the child. Every unneeded material, they also take it away from the child. And lastly, we have the embryonic membrane. the embryo while still in the womb has got all this membrane, while the amnion is underneath at the base. It prevents the baby in the womb whenever the baby wants to undergo shaking and all that. It protects the baby from shaking. It protects the baby from external temperature or excessive temperature. We have the chorion. The chorion protects those spaces where oxygen is coming in. They protect those spaces. They cushion where oxygen is coming. We have the allantois. The allantois is a membrane whose charge is to take care of respiration and excretion. We have the yolk sac. The yolk sac provides food for the embryo. And lastly, we have the umbilical cord. The umbilical cord connects the embryo to the placenta. Don't forget, the placenta connects the embryo to the womb. Are you with me? The umbilical cord connects the placenta to the embryo. You don't have time to say, I will have the mother. Okay, we have the mother. We have the embryo. What connects the two of them together is the placenta. Now, what connects the placenta and the embryo together is the umbilical cord. So lastly, we're going to be looking at the concept of oviparity and viviparity. Okay, so now we're going to be looking at two different concepts, oviparity and viviparity. Oviparity and viviparity. Oviparity are organisms that lay eggs. Oviparity are organisms that lay eggs. It is a concept used to characterize organisms that lay eggs. They lay egg. Their young ones are actually in form of eggs. So those eggs will hatch before they become like their parents. Are you with me? What viviparity talks about organisms that give birth to their young ones alive. They give birth to their young ones alive. They do not lay egg. They give birth to their young ones alive. So now we're going to be looking at the differences between these two different concepts. As I said earlier, oviparity lay eggs. Viviparity give birth to their young ones alive. For oviparity, okay, they lay egg, they do not. They give birth to their young ones alive, they do not. Fertilization in oviparity is external and internal. It is external for fishes. It is external for amphibians. It is internal for reptiles. It is internal for birds. For viviparity, it is only seen in mammals. And fertilization in mammal is internal. Fertilization in mammal is internal in the sense that it occurs inside. Don't forget, now the embryo develop outside the body of the organisms. They hatch their egg outside. Then they lay their egg outside, rather, the egg hatches. So the embryo develop outside the human body, outside the woman body. While for viviparity, the embryo develop inside the woman body. The woman becomes pregnant, the embryo develop inside the woman body. Examples of organisms that undergo oviparity are fishes, toad, frog, salamander, newt, birds, woodpecker, hummingbirds, and so on and so forth. Examples of organisms that undergo oviparity are man, dog, cat. Okay, so I'm taking this time out to explain the concept of oviparity, viviparity, and ovoviviparity. So look at it, we have ovi, vivi, ovovivi, ovi, vivi, ovovivi. So we're looking at these three concepts. Although it might sound funny, but it works the miraculous. Glory. So oviparity talks about organisms that lay eggs. They lay their egg. They lay their egg externally. Their egg will hatch and their egg will turn to chick. Are you with me? Just like we have our hen, the mother hen. The mother hen tends to lay egg, incubates the egg will hatch, and the egg will form another chick. So that's oviparity. Viviparity talks about organisms that give birth to their young ones alive. My mother, she got pregnant. I was in her womb, and she gave birth to me. I wasn't an egg. I did not hatch. She just gave birth to me, and I became a part of her. We share similarities and differences as well. So that's viviparity. Examples are mammals. Mammals are viviparous in nature. Oviparity, we have lizard, we have birds, we have fishes, we have amphibians. And now what is ovoviviparity? Ovoviviparity is a special concept. It talks about an organism, it talks about organisms that would not lay egg directly. Listen up, these organisms, they are going to lay egg. But they don't lay their egg outside. Their eggs are actually inside them. The egg will hatch inside them. And when it is time for the egg to form chick, they lay the egg. Ovoviviparity talks about organisms that carry their egg for a long period of time, and they make sure their egg hatches inside them. And when it is time for the egg to come out completely out of its shell, they lay the egg. So that's where it is. So the major difference between ovoviviparity and viviparity is that this one is it is connected to the placenta. This one has no placenta. There is no connection between the embryo and the womb. Yeah, there's a connection between the embryo and the womb, so we call it placenta. This one has got no placenta. They just lay the chick. Now the egg is coming out as a chick. So they carry the egg for a long period of time. The egg hatches inside them, and then they give birth to the chick. So that is the trick. The egg stays inside of them. Instead of them laying the egg externally, and the egg will stay outside before it hatches. They lay the egg internally. The egg stays there, and when it's time for it to hatch, they give birth to the egg. So examples of ovoviviparity is seen in snakes. It's seen in snakes. It's seen in shark as well. It's seen in snakes. Snakes, that's how they do. Their egg will stay inside of them to hatch there. Then they just give birth to another snake. But it was actually in the form of an egg. There's no connection. There's no placenta there. And same with shark. It is actually it's been a nice time out with you. Some questions will pop on your screen. Try as much as possible to attempt them. Should you be having issues with what has been taught so far, you could as well go through the video over and over again. Thank you.